Hi all. So we will continue the discussion with the T lymphocytes. So we have seen what are B lymphocytes, what are the features and the major functions. Now look at the the next important lymphocytes, and rather this is the most significant cell in the immune system. These are the central cells playing a very crucial role for the development of a number of immune responses, whether it be B cell mediated, whether it be antibody dependent, whether it be cell mediated or humoral mediated. For all those kind of immune responses, a particular cell called T cell, or more specifically T helper cells, plays the central role. So we are going to discuss about T lymphocytes in brief. So the name itself, T stands for thymus derived. We know B was standing for bursa fabricius, from the birds where there is an organ called bursa fabricius where the B cells matured. Here the T cells are given the name T lymphocytes because the T cells, after synthesis from the bone marrow, releases into the blood, and from the blood they migrate into the thymus. So thymus is a specialized organ for the development of these T cells, right? So the T stands actually for the thymus derived, that is the importance of thymus. So thymus playing a very central, very crucial role, very vital role in the development of T cells, right? So within the thymus, this is not simply a site of maturation, but there are number of reactions, number of selection processes. Collectively we call it as a thymic selection occurs in the thymus. Already we have seen the structural features of the thymus when we discuss about the organs of immune system. That is one of the important primary organ where these T cells mature. So there is a process collectively we call thymic selection, by which the T cells which are capable of identifying our self molecules or the self MHC molecule in specific only will be allowed to propagate further. That means those T cells which can recognize and identify the self MHC molecules only will be allowed to further develop, right? At the other end, those T cells which are strongly interacting with the self MHC molecule or with self MHC peptide complexes within the thymus will not be further propagated, and they will be subjected for cell death. These two mechanisms are collectively called the thymic selection, or specifically it is positive selection and negative selection. So positive selection is for the propagation and further development will be followed. In case of negative selection, those cells undergoing negative selection will be eliminated. This is an important stage of any important stage in case of T cell development, because those T cells which are reactive to the self cells are eliminated within the thymus itself. This mechanism is what we call this is the basis for the very significant reaction in immune system or immune responses called the unresponsiveness or the clonal, we call it as the unresponsiveness of the immune system against certain self molecules, right? So this occurs in the thymus. So this is part of the central tolerance mechanism of the immune system, and by which the immune cells will tolerate the self cells, and this is the central tolerance occurs within the T cells or within the thymus for the T cells. And after maturation, the T cells will leave the thymus to the circulation. So initially after synthesis from the bone marrow, they released into the thymus, then they left to the from the bone marrow they released into the circulation, then from there they migrated to the thymus, and after maturation they again released into the circulation, and from there they usually migrate to the secondary sites or the peripheral tissues, and the rest we know their way where they interact with the foreign substances for the next level of differentiation and activation. And when we talk about the receptors, like in B cells, there was a receptor called B cell receptor. Here also we can see a specific receptor called the T cell receptor. So this is the receptor responsible for the interaction of T cells with the corresponding antigens, right? So the T cell receptor and the B cell receptor having certain similar features, but they are not unique. They are not the same receptors, but there are some features which are sharing, but there are certain major differences also. So let's see what are the important features of T cell receptor. So unlike B cell receptor, this is not an antibody molecule. That is the most significant difference between the T cell receptor and the B cell receptor. This is not an antibody molecule. That we have to remember. This is not an antibody molecule, but B cell receptor was an antibody, that was an IgM or IgD molecule, right? But it is given that even though it is not an antibody, they belongs to the same family called immunoglobulin superfamily. Both BCR and TCR belongs to the same immunoglobulin superfamily, even though they are not antibody molecule, right? Antibody is a molecule within the immunoglobulin superfamily. T cell receptor is also an immunoglobulin superfamily molecule, okay? And there are two polypeptide chains with a variable and a constant region at the exterior portion of this molecule. You can look at this image. There is a variable portion and there is a constant portion for the here it is, this is the alpha chain. For the alpha chain itself, there is a variable domain and a constant domain, and for the beta chain also there is a variable and constant domain, which is outside of the membrane, right? And then you can see a transmembrane region which interact with the membrane, and also there is a cytoplasmic tail, right? And when we analyze in detail, there are two varieties of T cell receptors. One is called alpha beta receptor or alpha beta T cell receptor, and the other one is gamma delta T cell receptor. And the alpha beta T cell receptor is the predominant type in humans, and mostly in most of the mammals. And so we will be further discussing about alpha beta T cell receptor. These are encoded by the alpha beta genes. And here this will give you a more detailed idea about the T cell receptor. This is the structure already we have seen. There is a variable, constant domain, transmembrane, and cytoplasmic tails, right? And these two are connected by disulfide linkages between these two polypeptides. And another important point we are now entering into the features of the T cell receptor and the interaction of this T cell receptor with antigen. How this occurs? Is it similar to that of a B cell receptor or not? Is there any differences, or is there any additional supplementary molecules or cells required or not, right? So the first point is that T cell receptor recognizes this. You have to be very consciously listen to these things. T cell receptors recognizes those antigens that is bound to particular classes of cell molecules only, right? That means T cell receptors cannot identify an antigen if it is presented as such, right? T cell receptor never identify a foreign substance alone, right? But T cell receptor recognizes antigens when it is bound to a class of self molecule. Clear? That is the first statement. Then, what are these self molecules? A group of proteins called MHC proteins encoded by a gene cluster called, why they are called MHC protein? Because they are encoded by a gene cluster called major histocompatibility complex. That's what MHC stands for, major histocompatibility complex, right? These are the proteins, these are the self molecules. So the T cell receptor need to need antigens which are presented along with an MHC protein. Only then the T cell receptor can identify. Right. You can see the image where there is a T cell, and the T cell recognizes the antigen. Here you can see the foreign protein, or this is the antigen. This antigen is recognized by the T cell receptor, but the recognition of this antigen require assistance by the MHC protein. Alright. So here it is clearly we have to understand that the recognition of the B cell receptor and T cell receptor are not of similar kind. T cell receptor cannot recognize a free antigen, but B cell receptor can do so. Right. So they can recognize and interact with the peptide antigens. That's also important. Most of the antigens identified by the T cell or the T cell receptor are of peptide antigens. Right. So these peptide antigens which are processed usually by a particular group of cell called APCs, or antigen presenting cells. Right. So T cell receptor require antigens to be presented on certain self molecules by a specialized category of cells called antigen presenting cells. Right. So these APCs will present these molecules on an MHC molecule. Right. So that particular complex is called MHC, antigen peptide complex. You can see here, this is the antigen presenting cell. So this APC will present the antigen on a molecule called MHC protein. So these MHC proteins are the self molecules. We can have a detailed look at MHC and APC. So here it is about MHC. There are basically there are three categories of MHC proteins. One is class 1, class 2 and class 3. Right. MHC stands for major histocompatibility complex. This is a gene cluster, and that cluster of gene consists of number of proteins, and the class 1 consists of proteins which are present on all nucleated cells. Right. Class 1 MHC proteins are present on all nucleated cells of the body. So for every cell there will be class 1 MHC molecule on their surface, but for class 2 molecule they are present only on specialized cells called antigen presenting cells, or APCs. So class 2 MHC molecules are present only on antigen presenting cells. So what are antigen presenting cells? That we have to see. Right. So antigen presenting cells there are important cells called antigen presenting cells within the body, and these are the cells. Let me go back. These antigen presenting cells are these antigen presenting cells are those cells which can digest and present the antigen for recognition by T cells. Right. There are three major types of antigen presenting cells in the body. They are macrophages, B lymphocytes and dendritic cells. Right. They process the peptide antigens and present the sequences of the antigens to the T cells for the recognition. Then further T cells will be activated, and of which the dendritic cells are the most potent APC, and there are many other features also to be discussed about APCs and MHC that we will discuss in detail later. Right. So that's about this MHC. So you can look at the structures also. This is about the class 1 MHC molecule, and this is about the class 2 MHC molecule. So this class 1 is present on all type of cells, but class 2 is present only on the antigen presenting cells, or APCs. Clear? So here now we are moving into the functional features of T cells. So here this image shows gives you an idea how important T cells are. T cells are involved in diverse functions. Actually, this represents the functional features of T helper cells. So T cells plays vital role in various responses of immune system. Like they activate monocytes, they can activate NK cells, they can activate B cells. They are associated with the development and activation of eosinophils, then macrophages, then they interact with, or the chemicals interact with the liver cells for production of acute phase proteins when there is an infection, and also they activate neutrophils, and all those reactions are regulated or are under the control of T cells, particularly T helper cells. So this is why we are from the beginning itself I states that the lymphocytes are central to most of the immune response, particularly T cells, right. So what are the other receptors in addition to the T cell receptor? What are the other receptors on T cell? One is CD3. CD3 is an important receptor on T cell. These are required for membrane expression of the T cell receptor and for signal transduction, just like the MIG alpha and MIG beta seen in B cell. Okay, the another one is CD4, which is a co-receptor found on T helper cell only. This is important. CD4 is important. These are present only on T helper cells, right. CD4 receptors are present only on T helper cell and can interact with MHC class 2 antigen complex. This is an important statement that you have to remember well. This CD4 is a co-receptor found on T helper cells. So you have to connect CD4 and T helper cell. So T helper cell carry CD4 receptor, and these CD4 will help in the interaction of the T helper cell with class 2 MHC antigen complex, right. So that is an important point for CD4 receptor. Then the next receptor is CD8. This is also a co-receptor but found on T cytotoxic cell. This is another type of cells that we will come to discuss with the subtypes T helper cell and T cytotoxic cell, and what are the features and all. So here CD8 is present only on the T cytotoxic cells, and this can interact with class 1 MHC antigen complex, right. So these are the two important receptors that helps to differentiate cells into T helper cell and T cytotoxic cells. Then other receptors like CD28, CTLA4, all other receptors are commonly found on T cell surface. They all helping in the signal transduction, interaction with the other cells, and also in the activation mechanisms. Yeah, you can see the other common receptors. CD4, CD8 already we discussed. Then CD2. Then about the function and the family of that receptor. All those things are given here. Okay, in addition to the common T cell receptor, all these are the other receptors and their corresponding ligand. CD4 interact with class 2 MHC, CD8 interact with class 1, then CD2 interact with CD58, right. Then other things are there, CD28 with B7 family. Already we have seen that B7 is a receptor found on B cell surface, right. In the previous presentation we have seen that B7 family receptors are present on B cell surface. So they interact with the T cells, right. So CD28 and CTLA4 are the receptors found on T cells, and these two are helping in the interaction of T and B cells. Then all the other receptors are there. So this will give you about a pictorial representation of the interaction. So here you can see about T helper cell, you can see the interaction here. This is the T cell receptor, and this is the class 2 MHC molecule, and this is the antigen. And here you can see the CD4. Then other receptors, additional co-receptors are given. And in this right hand side of this image, you can see the T cytotoxic cell, where you can see the T cell receptor CD3 complex and also the class 1 MHC. Here class 1 MHC plus antigen, then additional co-receptors. All right. This will give you a better idea about how these cells interact with antigen peptide complex, antigen MHC complex. Then about the subclasses, we are talking about the T cytotoxic and the T helper cells. So what are they? So based on the CD markers and other functional features, we have differentiated the cells into two major categories. They are called T helper cell and the T cytotoxic cells, right. So the T helper cells are the cells with a CD4 receptor. That is the important unique marker to recognize. And you can look at the image. This is the CD4 receptor in addition to the T cell receptor. Then so these are CD4 plus cells. The second category, second subclass is T cytotoxic cell or TC cell, and they are having a CD8 receptor. Right. So this is the CD8 receptor representation along with the T cell receptor. So this is a CD8 plus cell. So this is the basic classification and the brief discussion on that T cells could be divided into two categories. One is T helper cell, the other one is T cytotoxic cell. T helper cell is CD4 plus cell, and T cytotoxic cell is CD8 plus cells. Right. And other points, already we know that CD4 plus cells are able to interact with antigen MHC class 2 complex, whereas T cytotoxic cells are able to interact with class 1 MHC antigen complex. Fine. And this is the effector mechanism. You can see there, T helper cells will be activated on interaction with antigen. Right. Here you have to remember the antigen is not alone. Antigen will be presented by the class 2 MHC molecule, because this is T helper cell. So here you can see the image. The class 2 MHC will present the antigen. So the class 2 MHC is present on the surface of antigen presenting cells, on APCs, and the important APCs are dendritic cells, macrophages and B cells. Right. So this APC will present antigen on class 2 MHC, and this antigen class 2 MHC complex will be identified by the T cell receptor MHC complex. So this is a CD4 receptor, and then this activated that leads to the activation. Now this is an activated T helper cell. This activated T helper cell will secrete diverse cytokines. A number of cytokines are released by the T helper cell, and these cytokines are responsible for the effector functions. Actually, these T helper cell is not directly involved in any kind of immune reaction, but instead these cytokines will mediate a lot of reactions. That's why these cells are called T helper cells. Instead of directly involving in the reaction or in the elimination process, they are associated with facilitating the immune responses. That's why they are called helper cells. Now look at the other cell, T cytotoxic cell. This is called cytotoxic because these cells will produce, after activation, will produce chemicals which are toxic to cells. That's why it is cytotoxic. See, you can look at T cytotoxic cells are activated on interaction with class 1 MHC. Class 1 MHC molecule, and we know that class 1 MHC molecules are present usually on all nucleated cells, right? They are present on all nucleated cells. So how this class 1 MHC is responsible for presenting an antigen? The normal situation is that when there is a self cell, when our own body cell is damaged, just like when a cell is having a viral infection, for example, or the cell is having a tumor development. In such situations, what the cell do is, the cell will present a self antigen, a self peptide on the surface of the class 1 MHC. Normally a self cell will not present any antigen on class 1 MHC. But in these kind of conditions, when there is a viral infection or when there is tumor development, in such situations, what the self cell do is that it will present an antigen on the class 1 MHC molecule on the surface, right? And the T cell or the T cytotoxic cell will identify these antigens whenever presented with the help of the T cell receptor and the CD8 marker, CD8 receptor on the T cytotoxic cell, and this causes activation, and this activation leads to the development of activated T cytotoxic cell called cytotoxic T lymphocyte. Okay, cytotoxic T lymphocyte or CTL, and this CTL will secrete chemicals, and these chemicals in turn target the cell which presented the antigen, right? So the activated CTLs recognize and kill the altered self cell. That means the self cell which presented the antigen for activation of the TC cell will be targeted. right? And because of this, these cells, the cells which are presenting antigens to T cytotoxic cell on class 1 MHC molecule are called altered self cells. So these altered self cell are target for destruction, and that will be done by the cytotoxic cell. Right. So that's about this mechanism or the effector reactions of T cells. Hope you followed this one. So in brief, T cells, I'll repeat the important points. The T cells are the major cells of lymphocytes. They have a T cell receptor on their surface. The T cell receptor is an immunoglobulin family molecule, but it's not an antibody. Then there are two categories of T cells, T helper cell and T cytotoxic cell, based on some co-receptors called CD4 and CD8. T helper cell carry CD4, whereas T cytotoxic cell carry CD8. Right. Then T cell receptor cannot recognize free antigen, but instead they require antigens to be presented on self molecule. And these self molecules, the important self molecules are called MHC molecule. These are a certain group of proteins found on cell surfaces, of which class 2 MHC molecules are present on specialized cells called antigen presenting cells, which include macrophages, dendritic cells and B cells. So those cells will process and present antigens to the T helper cell, and the T helper cell will recognize with the help of CD4 and the T cell receptor, and then they will secrete chemicals, and these chemicals will do the rest of the functions. Right, we have a diverse number of functions. And on the other hand, the class 1 MHC molecules are present on all nucleated cells, and in a situation where there is a viral attack or there is a tumor development, in such situation the cell is damaged. In those conditions, the self cell will present a self antigen on the class 1 MHC molecule, and the T cytotoxic cell can recognize this class 1 MHC peptide complex on the surface of a self cell, and these self cells are called altered self cells. Right, and this leads to the activation of TC cells, and the activated cytotoxic cell is called cytotoxic T lymphocyte or CTL, and these CTLs will secrete cytotoxic chemicals. Right, a lot of cytotoxic chemicals are released. These chemicals will target the altered self cell, and that will be destroyed. Right, so that's about these T lymphocytes. Hope it's clear.